Finding large-scale motions in proteins can provide information on the flexibility or rigidity of parts of the protein, on domain movements, or give some clues to biochemical processes. Trajectories from molecular dynamics simulations can show the large-scale motions of proteins, but these simulations are not resolved in individual modes and they require a large amount of computational resources. So, in this video exercise, we will go through the low-mode vibrational sampling procedure in BioLuminate to determine and visualize the lowest vibrational mode of a protein. The vibrational modes are generated as a set of structures sampled at regular intervals along a full cycle of the vibrational mode. While the rotational and translational modes are discarded, the vibrational modes will be visualized by displaying the structures in sequence as a movie in the workspace. The example protein used here in this exercise is the structure of anti-HIV1 antibody 2F5 light chain, which corresponds to chain L from the PDB1TJH. Now this protein has already been prepared using the protein preparation wizard, which is required before performing the low-mode vibrational sampling calculation. You should also delete any waters and solvent molecules so that you are analyzing just the protein and its ligands if present. Once you have a properly prepared protein, we can start the exercise by going to Tasks, Low Normal Mode Analysis, Calculate. Now for this exercise, we'll just use the default settings. But here, the number of vibrational modes to visualize can be changed. Here, the number of frames per mode can be set, i.e. the number of frames to generate per vibrational cycle. Keep in mind though that this number should be divisible by 4. This is because each frame is a snapshot of the structure at a particular point in the vibration between the classical limits, and the full cycle of vibration is divided evenly to define the coordinates used for the snapshots. And here, the maximum vibrational amplitude can be set. This corresponds to the maximum displacement for the fastest moving atom. So for proteins, the fastest moving atom could potentially move a large distance if the motion involves a long loop, for example. But this choice is somewhat arbitrary, namely you might for example want to exaggerate the motions just to make them easier to see. Okay, now when ready, we'll click Run, then choose a job name, and then click Start. Note that the limit on the number of atoms that can be processed depends on the memory available on the machine. So with 4 GB of memory, that's about 5,000 atoms. So the job could take several hours depending on the protein. When the job finishes, we can import the results by going to the Visualize tab, clicking Browse, and then selecting the .com file. Here the results are added to the project as an entry group. Then just specify which mode you want to view here. Typically for structures with 5,000 atoms, an interval of 0.1 seconds is fine and will produce a reasonable animation. We'll make sure loop is checked so we can loop continuously through the frames and have the vibration go through multiple cycles. And then all we have to do is use the controls to play the animation. Now you'll notice in our example, the largest motions are happening out here in the 2 domains, whereas this hinge-like region would consist of the bending residues. This is a good result because it's actually consistent with X-ray data, which you can see over here. Here I've superimposed the original X-ray structure with another X-ray structure of anti-HIV1 antibody 2F5 light chain from PDB1U8K to show that these bending residues do allow for motion in the domains.